Hi everyone, welcome back to Useful Genetics. This is Lecture 1K, which is going to be a very short overview of the marvels of modern DNA sequencing. So we'll talk about what the technology differs, how it differs from what we used to be able to do, and the different things that we can learn using this technology. So back in the good old days, before about the year 2000, DNA sequencing was done on pure samples of DNA consisting of many identical copies of a single DNA molecule, single DNA fragment, usually. And the result of that sequence was, of that analysis, was one DNA sequence that represented the average sequence over the whole sample. Modern DNA sequencing, in contrast, can take a sample that consists of a mixture of many different DNA molecules, and it can sequence each molecule in the sample, giving many, many different sequences as what are called sequence reads. Modern DNA sequencing is also an awful lot cheaper than it used to be. Here's a graph showing the cost of DNA sequencing as it's changed between the year 2001 and the year 2013. And it's compared with the Moore's Law decrease in the cost of computer power, which goes down. I think Moore's Law has it that the cost of computer power falls by half every 6 months. So for the first, 6 or 7 years, the cost of DNA sequencing decreased dramatically, roughly at the same rate as the cost of computing power. And then, starting in about 2007, it plummeted. And now it's back to still decreasing dramatically, but only at about the rate of Moore's Law. So DNA sequencing has become very cheap. Well, cheap for science. Nothing in science is really cheap. Now, I'm going to describe 3 applications. And the first is sequencing not just a gene, but a whole genome. So for an organism with a relatively small genome, we can take a single sample tube with the entire genome broken into fragments and sequence all the fragments. And then by comparing the fragments, we can line them up and find the overlaps that allow us to infer the complete genome sequence from the assembly of these short reads. For a larger genome, we can do the same thing, but we'll need to use multiple samples. Now, a second application is that rather than sequencing one genome, we can sequence a mixture of DNAs from different sources. DNAs from all the members of a population, or from all the species in a community. For instance, you can scrape the bacteria off your teeth and extract the DNA from this mixture of bacteria and sequence it. This is a study called metagenomics, studying the phenomenon of the human, what's called our microbiome, the ecological communities of bacteria that live on us. And when these sequences are analyzed, they can be grouped into particular groups that tells you the kinds of organisms that are present in this sample, even though you don't have any way to directly study these organisms. The third application of DNA sequencing is measuring RNA abundances in a cellular tissue. We've known for many years that different genes are expressed at different levels, and there's different amounts of RNA for different genes in the cell, but we didn't have an easy way to measure this. We still don't have a way to sequence RNA, but we do have a very easy way to use an enzyme to make DNA copies of the RNA, and then we use our very efficient DNA sequencing method to sequence all of the complementary DNA, it's called, in the sample, which gives us a measure of the sequences of the RNAs in the original sample. We can then basically count the amount of RNA sequence that we get for each gene in the genome, and that tells us how much RNA is present in that cellular tissue from each gene. So this is a gene that's expressed at a very high level. This is a gene that's hardly expressed at all. So what we've done. We've talked about how the new methods of DNA sequencing can sequence many DNAs at once rather than just a single molecule, and they can do it far less expensively than the old methods did. This enables very efficient DNA sequencing to be applied to new problems. We can sequence whole genomes much more easily, for instance, than we sequenced the human genome when we started. We can characterize the genetic membership of ecological communities just by sequencing the DNA without actually knowing what the organisms are. And we can use DNA sequencing to measure the abundance of RNA in a sample, which tells us how strongly different genes are expressed in different tissues. Coming up next, we're going to switch to think about an evolutionary concept, the concept of homology, which is vitally important for many aspects of understanding genetics. I hope to see you there.